the complement system. And the complement system is a very important part of immunity, and it's often one of the very first mechanisms that gets activated when there's a wound or some kind of infection by a pathogen. So we're going to go through the whole different steps of complement, what complement means, how it works, and the important role that it plays in relationship to all the other aspects of the immune system. So complement is a protein, and there are over 30 different kinds of complement in the human body, and complement resides in the plasma, and in fact it represents approximately 10% of the plasma. So it's a visible and important part, certainly, and it's synthesized by different cell types. So it's synthesized in the liver, also in the GI and genitourinary tract, and of course the blood supply. So the complement system consists of several different types of plasma protein, and these proteins, as you can imagine, are continuously circulating in the blood supply. So they're almost like messengers, so to speak, or molecules that can be activated instantly. And once these molecules are activated, all kinds of chains of events occur as a result of the activation. So the activation process of complement, and it actually occurs in a series of steps like a cascade. The activation actually results in the destruction of pathogens directly, so the complement can directly destroy pathogens. Most of the function of complement is actually to activate and alert other components of the immune system, and it also collaborates. So it alerts other parts of the immune system, and it collaborates with other cells of the immune system to basically eradicate pathogens, but also promote healing in a wound. So there are 3 pathways that are related to complement, and these are consistent, and these are the types of questions that you would get on a microbiology exam. Often represent questions like what kind of infection or pathogenic attack would the complement respond to, and which pathway would it respond. So it's important to understand what types of organisms are influenced by these pathways and what these pathways actually do. So the classical pathway is the complement proteins that are activated by other proteins from especially acquired immunity. So when these complement proteins are in the presence of activated elements from acquired immunity, then they get activated. So we'll learn in other lectures what the acquired immune system response is all about. Now, the lectin pathway are complement proteins that are specifically arranged or configured to come into play or to activate in the presence of lectin patterns that are present on bacterial cell walls, and these bacterial patterns or lectin patterns are actually carbohydrates. Now, the alternative pathway are particular complement proteins that are activated in the presence of gram-negative bacteria and also certain fungal cell wall polysaccharides. So just to show you these lectin domains, now this is an example of a plasma membrane, for instance, of a eukaryotic cell or also from a bacteria, and you can see this particular pattern here is an integral protein that goes through the membrane, and then the upper part is a carbohydrate. And so the complement proteins in the lectin pathway are configured to activate in the presence of the patterns of these lectin carbohydrates that are embedded in the cell wall of certain bacteria. So the function of the complement system is to become activated upon infection or recognition. So in other words, by becoming activated, in a sense, they're actually recognizing, because they don't get activated in the presence of other things. They only get activated in the presence of these certain patterns that exist on bacteria, for instance, but also they get activated in the presence of other immune cells. And they're in constant movement, they're in constant circulation. So in a sense, they're almost like a series of guards, for instance, and they're constantly moving and around in the blood supply. So when there is infection, they're probably already on the scene. So they circulate in plasma, and they're inactive. They're inactive until they are in the presence of these patterns, and then they get activated. So when they get activated, then there's a series of these particular proteolytic reactions that yield bioavailable fractions or fragments, and the activation is immediate. So the complement is often the very first activity that occurs in the presence of an infection. And the way I like to teach about complement or to explain complement is to also talk about enzymes, because they work very much like enzymes. And we know, for instance, that enzymes are protein, and enzymes are very specific, and they only become activated in the presence of certain patterns. So here is a picture of an enzyme, and when it's inactive, it's basically inert. And then when we have, for instance, in the case of the enzyme, it's called a modulator. So when a very specific molecule lands on the shape, or actually lands here, and causes the enzyme to actually change shape, that enzyme then becomes active, and then it can participate in a biochemical reaction. So the complement is very much like the bacteria. So in the presence of these certain bacterial patterns and cells in the immune system, the complement will get activated immediately, and then it will set off a whole chain of reactions that then alert and cause other cells to come on the scene and actually begin the whole inflammatory response that occurs in the immune system. So complement is exactly what that word means. It complements, it works with other aspects of the immune system to eradicate these antigens and pathogens. But in some cases, the complement can actually destroy these pathogens directly. So it complements the ability of antibodies and phagocytic cells to identify and remove foreign pathogens. It also flags cells for what's called opsonization. And basically what that is that these complement proteins actually serve as flags that tell the immune system that this particular bacteria, for instance, requires eradication. And then they recruit these granulocytic other white cells or other cells from the immune system, and they attract these cells to the particular bacteria and then flag it for destruction. So with all of this activation that's going on with complement, that leads to an amplification of these immune reactions, and such that eventually we have death of the foreign cells. And also the complement facilitates the formation of these membrane attack complexes, which then leads to the direct destruction of cells. So here we have 4 different outcomes of complement activation. So the first outcome is lysis of the cells, which is direct destruction of the cells, such as bacteria, virus-infected cells, and tumor cells. Now, chemotaxis, what that word means is it means being attracted to something or to cells because of some kind of chemical that's secreted. So chemotaxis is attraction based on a chemical attractant, so to speak. And it's a process in which an immune cell, okay, a phagocyte, for instance, is attracted to a particular area of the body, and it moves towards a particular factor. And with complement, one of the outcomes of complement is it creates this chemotactic situation where all these cells, all these immune cells, are attracted to that particular area where the pathogen has entered the body. So here we have this figure of these two particular pathways, the classic and the alternative, and it just shows you the different ways in which they work and what ends up happening as a result of all of them. So the other two outcomes, opsonization, which we talked about, and that's where you actually flag the organism, the bacteria, for destruction. And basically the bacteria is coated with molecules, and that these molecules are then phagocytes actually are attracted to these molecules, and then they can eradicate or phagocytize, is the word, these identified cells. And anaphylotoxin is a particular situation where we have vasodilation and permeability changes that happen as a result of an infection, of course, and we have inflammation. So this inflammatory process is actually allows these cells to migrate in to the tissue, for instance, from the blood vessels, and it even allows more complement, antibodies, and immune cells to work towards eradicating the pathogen. And it serves as an inflammation initiator. So basically, complement is pretty much, when I teach immunity in immunology, I always talk about complement as being one of the very first things that happens in the immune response. So here is a picture of the formation of this membrane attack complex. So you can see that these are all these different types of complement proteins, and they actually tunnel in to the membrane of a bacteria, for instance, and they eventually form a channel. And that channel then, once the protein, different complement proteins encircle this whole area, we get a formation of a channel or a transmembrane pore. And that transmembrane pore is where we can get extracellular fluids that then cascade or pour into the cell, and then we get cell destruction as a result. So this is one of the ways in which complement can directly destroy a pathogen. So there are three. So here are the three pathways presented slightly in a more complex way. Now, most courses that you're going to take, either in microbiology or medical immunology, generally don't require that you understand all the different individual complement proteins, but rather that you understand the function of the three particular pathways, what kind of pathogens are most affected by these pathways, and what do these pathways actually accomplish. So again, to review, the classical pathway is the pathway that interacts with adaptive immunity, the antibodies and the T cells. It's dependent on antibody. The lectin pathway, remember, is the pathway that these complement proteins are activated in the presence of these lectin carbohydrate patterns on the bacteria. And then the alternative pathway is the complement proteins that are activated by gram-negative bacteria and also some fungal proteins. So in other words, when these pathogens are present or enter the blood supply or enter the plasma, so to speak, then these complements become activated, and then the whole process starts. So here are our questions that we always have at the end of our discussions. So the first one asks, which complement pathway identifies bacteria on the basis of carbohydrate patterns? So we talked about that quite a bit. The answer to that is called the lectin pathway. And the lectin pathway is based on activation of complement due to lectin. Now, what was lectin? What did lectin mean? Remember, it was a particular kind of configuration of a carbohydrate. Okay, so lectin means particular configuration of carbohydrates. So basically, the lectin pathway is the complement activation in the presence of lectin pathways or lectin patterns on the bacteria. Now, the second example has to do with which complement pathway interacts with antibodies and antigens. So again, the best way to answer these questions sometimes is to have an understanding of all three, and if at least you understand two of them, you can eliminate the third. So we've already talked about the lectin pathway. Okay, now the other pathway, remember, the other pathway that's determined by patterns of organisms is the alternative pathway. Okay, so the only other one left is the classic pathway. So the answer to example number 2 is the classic pathway. Okay, so the third question has to do with the question of opsonization. Will you get a question on this? Absolutely. Opsonization is a very important concept, and remember, the membrane attack complex was a little bit like that, but also, here's our bacteria, and if we end up putting flags up there to tell the immune system where is this particular bacteria, boy, we're going to get an attraction of quite a few cells on the same, and then those cells will actually eradicate the bacteria. So that concludes our lecture for the day. Thank you so much for visiting educator.com.